Currently, the chemical manufacturing of essential chemicals is harming our environment substantially. It not only eats up a lot of costly resources, but also releases toxic side products into the air. Imagine if we could produce such desirable compounds just from sunlight. To tackle this question, we are currently taking mosses from the nature, growing them in the lab, and evaluate their potential as an alternative for the synthetic cell factory for the production of fine chemicals. Our mosses can be grown in liquid cultures and bioreactors. You can compare these cell cultures with an industrial factory pipeline on nanoscale. Those factory pipelines have different valves controlling the conditions like temperature and oxygen, as well as supplying substrates in a way that maximizes the productivity. In our moss cell cultures, conditions, but also molecular regulatory mechanisms such as gene expression decide upon productivity. My aim is to try identify limiting genes that can be used as such valves to supply the cell more of the things it needs to produce our fine chemicals. So by understanding the limiting factors, we aim to find ways to increase the productivity of our moss cells for the production of natural compounds. That way, we want to get one step closer towards a more sustainable production platform for fine chemicals from plants.